recognition of your body's orientation in its environment and discrete movements of your body in its environment. Or maybe movements of your body as your environment moves. Basically, equilibrium is just the recognition of your body's orientation. And this helps you with things like balance. Because if your body's moving in one direction too much, you could fall over, right? But because your body recognizes that you're becoming unbalanced or out of its normal equilibrium, you have skeletal muscles that can contract and can bring you back to proper orientation, right? So how does your body actually sense that you're getting out of this equilibrium? Well, your inner ear does that. Your inner ear is, like I said before, much more complex than originally we kind of drew it out. So let's draw another little simple schematic of the inner ear. We can get rid of your cochlea real quick, because we've already covered it. So we know there's a cochlea, and this is the one part of the inner ear that's important for sound perception. But we also have this big chamber that the cochlea opens into that we call the vestibule. And the vestibule is going to be a part of balance, all right? Or part of equilibrium recognition. And you also have some weird little canal-looking things. I'll kind of draw them a little more appropriately. That we call semicircular canals. Let's see, so get my orientation down right here. These things called semicircular canals. That are also going to be involved with equilibrium, all right? Now, understanding these each, we can kind of go over the vestibule and the semicircular canals separately, all right? So each of these little chambers, canals themselves in this vestibule, have membranous chambers inside of them. So this is bony material, just like the cochlea. But remember, if we opened up the cochlea has a soft snail membranous structure inside, right? Our little snail inside of there, which terminates blindly. Then we have our little oval window, which the stapes communicates with, our round window. So we have a membranous, basically, sac inside of that cochlea. That's the same thing with the vestibule. You have a couple different sac-like structures inside of there. You have one that we call the utricle. And one that you call the sacule. Sacule, so two different little membranous sacs inside this bony vestibular matrix, right? Now the semicircular canals also have sac-like structures inside of them. So all those would have little sac-like structures inside of them as well. All right. So let's talk about the utricle and sacule first. The utricle and sacule are pretty easy. It's sort of semicircular canals because they all work exactly the same. But notice how I drew the utricle and sacule perpendicular to each other. All right. I drew them perpendicular to each other. This is going to be important because if I drew somebody, let's draw a little person here. accelerating toward you, how do I make an arrow so it looks like it's accelerating toward you? Here, how about go with this here? If we have this little guy accelerating toward you at a rapid speed, or accelerating backward at a rapid speed, what starts happening as you accelerate forward? Your body starts leaning backward, right? And if you didn't have proper muscular contractions in your core, you would fall over. So how does your body recognize to stimulate these types of contractions so you can stay standing upright? When you're accelerating forward, or in reverse, your saccule recognizes these types of movements. When your body's accelerating side to side, that's saccule. Your utricle recognizes these types of movements. And it's because of the movement of fluid inside these chambers. And it's going to be very similar to hearing in terms of your cochlea. You're going to have little tiny hair cells that are going to move in the opposite direction that fluid is moving. And those little tiny hair cells, when the cilia are moved in one type of direction, it's going to cause either a receptor potential to be generated or not generated, which can then either stimulate or not stimulate an action potential in afferent sensory neurons, which can be carried to the temporal regions of your brain, giving your body or your brain primary sensory information on what's happening to your body in terms of its movement orientation. So how does this all work? It's actually pretty simple, but you have to know some terminology about these different chambers. So let's just focus on one right now, since they work exactly the same. So let's draw we'll use a utricle. So the utricle itself, just like, you remember our basilar membrane, how it was lined with epithelial, or not really epithelial cells, nervous cells, but it was lined with hair cells. The entirety of the utricle membrane is also lined with hair cells. Our little hairs on there. All right, there's our little hairs. Now, unlike our basilar membrane in the cochlea, there's actually some other cells that are associated with these hair cells. We have cells called support cells. You usually draw these in green. I don't know why. Let's draw them in a different color this year. Let's go with black. So in between these hair cells, you have these cells called support cells. We're going to make them really ugly looking. We're not going to draw nice little support cells. This is easier. Accurate as possible. All right. So we have support cells, and we have hair cells. Just like in the cochlea, these hair cells act as separate cells. So separate cells. So just like in the cochlea, it's these hair cells that are responsible for producing receptor potentials. Now these support cells are not involved with type of potential generation. We'll talk about what they do here in a second. Now, every single little hair cell also forms a synapse, kind of like our little organ Corti cells of the cochlea. Oh, we can draw circles, that's easy. All these little hair cells form synapses with afferent sensory neurons. Lots of afferent sensory neurons. So we have afferent sensory neurons. And these are the cells that actually produce the action potential. So these first order sensory neurons that produce action potentials in response to whether or not receptor potentials are being generated at the hair cells. So again, very similar to the cochlea, which was very similar to the retina. If those photoreceptors in the retina were separate cells, that whether or not they were producing a receptor potential basically resulted in whether or not you get an action potential stimulated in one of these downstream afferent neurons. Same thing with the cochlea, the hair cells of organ Corti, and now same thing with the utricles and the saccules with the hair cells that line those membranes. All right? So, same basic principle. Now, all these neuron axons eventually come together to form a specific nerve. They form something called the vestibular branch. Of the vestibular cochlear nerve. So once you learn your brain cranial nerves, you might have one called the vestibular cochlear nerve. Did you guys learn that nerve yet, that cranial nerve? Sometimes they used to just call it the auditory nerve, because there's a vestibular branch, and then there's also a cochlear branch. So this cochlear branch is basically formed through the neurons that are leading away from the cochlea. So they form a common nerve after they leave your inner ear apparatus. So we used to just call it the auditory nerve, but I think you're probably being taught of the vestibular cochlear nerve because it's a little bit, makes more sense if you call it, it has two different functions, right? Like two different sensory functions. So, we've got our, let's see where we are so far. We have our utricle, we have our hair cells, we have our neurons, and we know what the hair cells are going to do, we know what the neurons do. These hair cells produce receptor potentials, and just like hair cells of the organ Corti, it's going to be whether or not fluid or some type of substrate moves over those hair cells, causing them to move. Now this is where we get a little bit different, because remember in the cochlea, we had endolymph. So it had fluid very similar to cerebrospinal fluid moving over those hair cells. So that was a very fine fluid viscous mixture. Now, in your utricles and saccules, you don't have endolymph. That endolymph is actually replaced with a substance that we call the otolithic membrane, at least right around those hair cells. So basically, you have something called you have this